other questions we can address? I will address the antigenicity of the molecules for sure. That is very, very important, so definitely going to address that. Any other questions? Give me a second. Okay. So if there are no other questions, let me just have a quick rundown of chapter 2. One of the things I do with chapter 2, which it is addressed in the PowerPoints, is that I don't start the chapter from the beginning, because I assume that you already know the basics of the chapter. You already know what are molecules, etc. So what I do is I started, and I'm trying to see if I have, no, I don't. I started from the point where it begins talking about organic molecules, which is the 2.4 in the ebook is section 2.4. I know if the, I guess it would be chapter, page 29 or 30, something like that. It doesn't correspond with what the PowerPoint says. The PowerPoint is outdated in that regard. So it's section 2.4 when it starts talking about organic molecules, and that's where we essentially begin the chapter. That's one thing. The other thing is that we do from the beginning talk about antigens and antibodies. Even though the chapter doesn't address that, I do start addressing it because it is so important. And because even though the book doesn't explain it until much later, it talks about it. Almost starting chapter 3, maybe we'll start talking about antigens, and it doesn't even explain what an antigen is until later on in chapter 15. So that's why I like to start on it as soon as possible. So the chapter focuses on something that you should already know from Biology 1406, the 4 basic macromolecules: proteins, carbohydrates, lipids, and nucleic acids. However, we're going to look at them from the lens of microbiology. So keep that in mind. These are the molecules that make up living things. And the idea about these molecules is 2 things. First of all, living things are made of these molecules. Number 2, these molecules are made by living things. So as far as this planet is concerned, all living things are made of carbohydrates, lipids, proteins, and nucleic acids. And the only way we find these molecules is going to be by living things making them. Okay, so you don't find proteins in a rock unless there is a living thing in that rock making those proteins. Okay, so I just want to emphasize that those molecules are indicative of life because only life can make them. Living cells can make them. They're complex. This is just going over where we find them, examples of locations where these molecules are found and where they're made. And notice that this is true for both eukaryotic and prokaryotic cells. Both cells contain these molecules. These molecules are huge. That's why they're called macromolecules. They are organic, meaning they have carbons attached to hydrogens in them. They are huge, like I mentioned. And they're polymers. They're made by smaller components. So it's not that they are one huge molecule, but they're actually polymers, or it's made of smaller pieces put together. So like if you think of a Lego tower made of smaller little Lego pieces, which makes the big tower. Same thing here. These are polymers. They have small pieces that make up the large molecule. And for the most part, each of these molecules each type of macromolecule has a specific polymer, a specific, piece that makes them up. Okay. I don't know what that is. All right, sorry about that. I'm going to turn it off. Okay, so what else? Okay, so you should have seen dehydration, photosynthesis, and hydrolysis reactions in Biology 1406. So just review those. Dehydration synthesis is what makes these molecules within the cell. So in a ribosome, for example, a dehydration synthesis will be making amino acids. In let's say in the cytoplasm of cells, you would have hydrolysis reactions going on that would break up, let's say, carbohydrates, for example, into the individual glucose molecules, for example. So these molecules are being broken up to get their component parts, or they're being synthesized from component parts. So review dehydration synthesis and hydrolysis reactions, and be able to recognize examples of dehydration synthesis or hydrolysis reaction. So here you have the first one is a dehydration synthesis, because you have two monomers, glucose and fructose, that are going to be bonded together. Notice that the process requires energy, and it also requires the addition of a water molecule. I'm trying to make my pen work here. So when you input energy into the system and remove a water molecule, you will bond these two monomers together, glucose and fructose, and now you have a new molecule called a sucrose. By the same token, sucrose could be broken down into its individual monomers, glucose and fructose, by the addition of water. And because we're breaking up chemical bonds, now we're going to release energy. And now we have the little components back out. All right. Feel free to interrupt at any point if you need to ask questions. Inorganic versus organic molecules, again, you should have already seen these in Biology 1406. Basic of it is inorganic molecules have no carbons, or if they have carbons, the carbons are not attached to hydrogens. So CO2 has a carbon, but it's not attached to a hydrogen, it's attached to an oxygen. So that is considered an inorganic molecule. H2O, for example, has no hydrogens, no carbons, so it is an inorganic molecule. Organic molecules have carbons, and they are attached to hydrogen. So that will be an organic molecule, for example. Okay. So be able to distinguish organic versus inorganic molecules. These next PowerPoints are just pointing out things that some of you may already know if you've taken chemistry. You may not know if you haven't taken chemistry. So I just thought I will go ahead and throw it in there so that we're clear when we go into metabolism chapters. You are more comfortable with these depictions of molecules, and you understand what they mean. So this is a glucose molecule in a linear configuration, but oftentimes sugar molecules are in a ring configuration. And in a ring configuration, notice that here the molecule has 6 carbons, 4, 5, 6. In the ring configuration, you only see 1 carbon out here outside the ring. The reason why is because chemists have their own shorthand, if you will, in which in the rings, every time you have a line coming together, a corner, if you will, you assume there is a carbon there. So there's a carbon right there. Here there isn't a carbon, there's an oxygen. That's why they actually put the little O for oxygen there. There's another carbon here, there's another carbon here, there's another carbon, and then that's the 5th carbon, and the 6th carbon, because it's outside the ring, is actually written as a carbon. So within the ring, the carbons are not written. Instead, it is assumed that every corner has a carbon. And if it doesn't have a carbon, it will tell you what it has. An oxygen, sometimes it's a nitrogen, so you'll have an N there. But if there isn't a letter, the corner is a carbon. Carbons are numbered, and we'll see that in chapter 7 when we kind of look at DNA. And they're typically numbered from the molecule in the ring that is not a carbon clockwise. So this is an oxygen. So from that oxygen clockwise, this is carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and outside the ring is carbon 6. So this is a glucose molecule, which is a 6-carbon compound. All right, now antigens and antibodies. Antigens are molecules, okay? And these are molecules that generate the production of antibodies in animals that have backbones, in animals that have an immune system with a backbone in it, with a bone marrow that is going to make immune system cells that are able to make antibodies. So, give me just a second. I'm looking for the PowerPoint that has the definition of antigen, and maybe I don't have it. Let me see. Give me just a second. Oh, there it is. So an antibody is, an antigen is a molecule. Now it says substance. To be more specific, you can say molecule. That's more specific than substance. It's a molecule that triggers the production of antibodies in an animal that has a backbone. So that's why it's called antibody generating. That's where the name comes in. So notice that the definition does not say anything about the molecule being foreign to the animal. It just says it's a molecule. It doesn't say it has to be foreign, because it doesn't. Okay. Now, there are two rules for a molecule to be antigenic. One is that the molecule has to be large. The other one is that the molecule has to be varied. It has to be complex, if you will. Okay. So a molecule that is repetitive, doesn't matter how large it is not antigenic. The molecule has to be large, and it has to have complexity to it. It can be a repetitive carbon, hydrogen, carbon, hydrogen, carbon, hydrogen, carbon, hydrogen. That's not antigenic. Okay. That doesn't stimulate the immune system. It needs variety in its structure. So those are the two rules for a molecule to be antigenic. Notice that antibodies are proteins, which are large, complex molecules. So that tells you immediately that antibodies could act as antigens in an immune system. So when an immune system is exposed to an antibody that is foreign to that immune system, the immune system will create antibodies against an antibody. Okay, so that's an important concept too, because we use those in microbiology to identify antibodies. We use anti-antibodies. So antibodies are antigens, or could be antigenic, I should say, in an immune system that is not the immune system that made them under normal circumstances. Okay, hopefully that's not too confusing. Main thing here, antibodies are molecules, large complex molecules. So the next question is, as we go through these macromolecules, lipids, amino acids, proteins, nucleic acids, and carbohydrates, which of these are large and complex enough to be antigenic? And why do we care? Why do we care if these molecules are antigenic? Well, the reason why we care is because a bacterial cell may have these molecules on their surface. So remember that bacterial cells have cell walls. What are the cell walls made of? Are the cell walls made of large complex molecules that could be antigenic? And if that's the case, is our immune system going to mount an immune response against the cell wall? If that's the case, could we use cell walls as vaccines? If that's the case, could we use cell walls in an antigen-antibody reaction to identify the organism? Bacteria have proteins sticking out of their cell walls. Are they antigenic? Some of these are receptor proteins, for example, or channel proteins. Could they be antigenic? The flagella of the bacteria is proteins. Is that antigenic? And if it is, could we use it to identify the bacteria with an antigen-antibody reaction? That's the reason why we address these antibody, antigen, or I guess antigenicity of molecules so early on, is because that's what we use oftentimes to identify bacteria, to make vaccines, to understand how the immune system responds to a bacterial threat. Okay, so as we go through these macromolecules, we will point out which ones are large and complex enough to be antigenic. So we start out with proteins. One thing you need to do for each one of these macromolecules is to know their building blocks. What are these large molecules made of? In the case of proteins, these are amino acids. Do not confuse amino acids with nucleic acids. Okay, so be careful with that. Amino acids are smaller building blocks made of three components: carbon, hydrogen, oxygen, and nitrogen. And they're made in the ribosomes of cells, and they are made as long chains of these amino acids put together. The long chains, if they become more than 50 amino acids put together and folded into a complex structure, then we call it a protein. However, oftentimes the cell is just going to make a chain that is long, but it's not 50 amino acids, it may be 20, maybe 4. We call those polypeptides. They're not complex, they're not proteins, they're just short chains of amino acids. Sometimes the cell may use several of these polypeptides to create a protein. Okay, so keep in mind, when we're speaking of protein, we're speaking of large chains of these amino acids. If we talk about a polypeptide, we're talking about a smaller kind of chain that is not considered a protein because it's not large and complex and folded in a specific structure. It's just a small chain of amino acids. Okay, so there's a bit of a difference there. Okay, function. I know this is a scrambled PowerPoint, but it's not scrambled in your computers when you look at it. The beauty of proteins is that when it comes to function, you can answer anything and you're correct, more likely. Any function in the cell, doesn't matter what it is, will probably involve a protein. However, having said that, the function that I would like to emphasize is that of enzymes. Proteins act as, some proteins act as enzymes, which are biological catalysts that speed up reactions. So that is the most important, well, most important, one of the most important functions of some of these proteins. The other thing about proteins that are important to keep in mind is that their shape and function are linked. The protein shape determines its function, and its function is dependent on its shape. So if the protein is supposed to be a channel, remember, it starts out as a chain. That chain has to fold into a channel for it to function. If the shape is returned back to its original linear shape, the protein stops functioning. So shape and function are linked in a protein. As I mentioned, proteins are made of amino acids. There are 20 naturally occurring amino acids, and the basic structure is the same for all of them. What changes between them is one of the groups attached to a carbon in the amino acid is different from each of the 20 different amino acids. So the basic part of the amino acid is the central carbon attached on one side to what is called an amino group, which is a nitrogen attached to hydrogens. On the other side, what's called a carboxyl group, which is a carbon attached to a hydroxyl and a double bonded oxygen group. The third arm of the carbon will have 4 free electrons to attach to 4 different chemical groups, is hydrogen. The fourth one is the one that varies among all 20 amino acids. So these are examples of all the 20 amino acids. The boxes in the brownish, the tan color, is the basic structure, and this is the same for all 20 of them. What changes is the blue box, which is different for every one of the 20. Having said that, all 20 could be put into different groups. Some are nonpolar, meaning they don't have a charge, an electrical charge. Others are charged, so they are polar. So you can see the negative charges in the oxygen. Others are, I'm gonna read there what it says, uncharged also. Some are hydrophobic, some are hydrophilic. So that is basically the classification. They can be classified as being acidic, basic, charged, not charged, hydrophobic, hydrophilic. So they have different properties. Okay, there is just a little 5 of the 20 amino acids. And by the way, you should be able to recognize a molecule as an amino acid. The way you do that, all of them are going to have that central carbon. All of them are going to have nitrogen. The the only two macromolecules that have nitrogens are the proteins and the nucleic acids. Lipids and carbohydrates do not have nitrogens. So you know that if there's a nitrogen somewhere, you're looking at either a protein or a nucleic acid. Nucleic acids are huge, easy to distinguish from a protein. Okay, the bond that brings together two amino acids is called a peptide bond, which is obviously formed by a dehydration system, synthesis. And then the other thing about proteins that you have to remember is that they're made in the ribosomes into these chains. So the peptide bonds bond by covalent bonding, which is a very strong bonding, bonds these individual amino acids together into a chain. The chain almost immediately within the cell as it's being made begins to twist and bend into alpha helices and or beta sheets, pleated sheets. And so now this huge chain has a secondary shape, which is the either an or alpha helix beta sheets. I said an or because some of these chains are so huge that they may have part of it an alpha helix and then another part would be a beta sheet, for example. So it's not one or the other, it could be both. And by the way, these structures are created by weak, easily broken ionic and hydrogen bonds, as opposed to the primary structure, which is not easily broken. Tertiary structure, easily broken, weak ionic and hydrogen bonds. Now we have these beta sheets and alpha helices twisting again in a three-dimensional structure. And that is typically the end of the shape of the protein. In some proteins, we have 2 or 3 or 4 tertiary structures coming together with weak chemical bonds to make what's called a quaternary structure. Okay, so these would be huge molecules. Now, notice that quaternary, tertiary, and secondary structures are made by weak ionic and hydrogen bonds. These bonds are easily broken by changing the environmental conditions. So if we change pH, temperature, salt concentration, these bonds will break and the protein begins to unfold. We call that the denaturing of the protein. And so proteins are fairly easily denatured back to the primary structure. Now, we could continue on with the extreme conditions and denature the protein all the way to the individual amino acids. That is absolutely possible, in which case there's no going back at that point. These could maybe be reversed if the conditions subside, but once you denature all the way to individual amino acids, there's no going back. So you should know what we mean by denaturation of proteins. We do that all the time when we heat an egg, for example, and we cook the white of the egg. It's very quickly, cooks extremely quickly, quicker than the fat, very sensitive to the high heat. And once you have it in that solid white form, there is no going back to what it looked like before. Okay, so another characteristic of proteins which is important is that they can attach to other compounds very easily. So they can attach to sugars, lipids, phosphorus, and so we have these complex proteins, glycoproteins, lipoproteins, phosphoproteins. So these can be not only large and complex molecules on their own, but on top of that, they can make themselves even more complex by attaching to other compounds. What that tells you is that proteins are not only antigenic, but they're the best antigenic molecules. There's nothing better than a protein when it comes to antigenicity. So if you want to induce an immune response, you want to use a protein because that is the best antigen out there. So that's the basics of the protein. The next molecule that is looked at are the carbohydrates. And there are different ways to approach them. None of them are necessarily better or worse. I'm going to show you the way I like to approach them. So first thing about carbohydrates is that, yeah, the names often end in the OSE suffix. That's some of the root words used for sugar or glucose. They are made of carbon, hydrogen, and oxygen. Notice there is no nitrogen. So the building blocks are called monosaccharides. And in contrast with proteins, which have the building blocks are called amino acids, and there are 20 of them in the naturally occurring world. When it comes to monosaccharides, there aren't that many. You can almost argue there may just be 4 or 5 that are mostly used by cells. You can even argue there's one that is the one that is mostly used by cells, which is glucose. So there isn't the variety, hint, hint, that you get with proteins when it comes to building blocks. So the main monosaccharide is glucose, which is a 6-carbon monosaccharide, also called hexoses. There are other monosaccharides that are 6 carbons, like fructose and galactose. So those are the 3 most common 6-carbon monosaccharides: fructose, glucose, galactose. And yes, you do need to know that. Again, those are the molecules in ring and linear form. Okay, we got that. There are a couple of 5-carbon monosaccharides that you do need to know because they will come back. They're important. These are called ribose and deoxyribose. And these are the monosaccharides that are used in DNA, in nucleic acids, DNA and RNA. That's what you need to know them. Those are the sugars, if you will, found in DNA and RNA. 2 monosaccharides come together to make a disaccharide. Disaccharides, common disaccharides: sucrose, which is table sugar made of glucose and fructose; lactose, which is the sugar in milk made of glucose and galactose; maltose, which is made of glucose and glucose. Okay, so know these 2 and know what they're made of. And then these monosaccharides could make 3 carbon sugars or 4 carbon sugars or 10 carbon sugars or 20 carbon sugars. This is what we call sugars. Sugars are the small chains, if you will, of monosaccharides, even though some of them could be rings, but okay, chains of monosaccharides. So a small would be, like I said, 2, 3, 4, 10, 15, 20. Those are small sugars. The key with sugars is not only that you have a small chain of monosaccharides, is that sugars are sweet and they dissolve in water. The longer the chain, the less sweet and the less they dissolve in water. So that as these chains become hundreds and hundreds and hundreds of monosaccharides put together, now we have polysaccharides. Sometimes they're called starches. I don't particularly like to call them starches because a starch is a particular type of polysaccharide. So I just like to call them polysaccharides because that encompasses all of these large 100 chains or more of monosaccharides put together. Most, by far, not to say all, but most of these polysaccharides are going to use glucose as the building block. So that all you're going to have is chains and chains of hundreds and hundreds and hundreds of glucoses put together. The most important polysaccharides: cellulose, glycogen, dextran, starch. Okay. The glycogen is the storage form of energy for both animals and bacteria. The starch is the storage form of energy for plants. Cellulose is the most abundant polysaccharide in the planet and is what makes up plant cell walls, the bark of trees, etc. Algae cell walls for the most part also. A dextran is a polysaccharide that is exclusive to bacteria, and this is a sticky polysaccharide. Look at the processed foods you may have in your pantry for dextrans, which are sugars, these polysaccharides that are sticky. They're made by bacteria, and they're used to bind the components of the processed food together so that they don't fall apart, they don't disintegrate. So this is a picture of polysaccharides. Each one of the little circles represents a glucose molecule. And there are two different polysaccharides. One is glycogen, the other one is cellulose. And notice that they are basically made of the same monomer. What changes is the way these monomers are bonded together, creating different shapes of these huge molecules. So the question would be, you have these huge molecules that are made of repeated glucoses. Could they be antigenic? Do they pass the test for antigenicity? Are they large? Yes, they are. Are they complex? No, they're not complex. They're made of the same molecule repeated over and over again. Now, one thing about these polysaccharides is that they also have the ability to bind with other molecules, like amino acids or lipids. And then I like to call them then complex polysaccharides when they're bound to something else. So, and actually the book also calls them complex polysaccharides. So a complex polysaccharide would be a polysaccharide with something else bound to it. Now, once you bind something else to the molecule, like for example, these NAMs and NAGs, these are essentially glucoses. It's modified glucoses. This is peptidoglycan. Bacteria take glucose, modify them a little bit, and create their own unique version called N-acetylglycolic acid and acetylglycosamine. These are essentially glucoses, modified glucoses, which are being used to create these long chains of very boring NAM, NAJ, NAM, NAG, NAM, NAG. So yes, they're large. No, they're not complex enough to be antigenic. However, to each NAM molecule, 4 amino acids are attached. Now we have a complex polysaccharide. This is antigenic because of this new molecule being attached to the very boring polysaccharide, making it a more varied, antigenically speaking, more interesting molecule. So, complex polysaccharides are antigenic. Polysaccharides are not. Starch is not antigenic. The starch bound to a protein, now we have an antigenicity compound. Go over the functions. I think you should already know what the functions are. Complex polysaccharides are antigenic. Polysaccharides by themselves are not. Having said that, they're not as good of an antigen as a protein would be. So they are antigenic, but they're not great like a protein. So between choosing a complex polysaccharide versus a protein to make a vaccine, for example, something that would have to stimulate the immune system, you would choose a protein if you can. Sometimes you can't. Sometimes you have to use a complex polysaccharide, and then you have a problem with the vaccine not being very antigenic. Again, if anybody has questions, let me know. The next molecule we look at are nucleic acids, and these are going to have their own chapter. Chapter 7 is going to be just on nucleic acids, so we're not going to spend too much time on them. First thing is to know the building blocks, which are called nucleotides, and to know there are 2 types of nucleic acids, the DNA and RNA. Both of them are made by essentially let me go back. Nucleic acids are made of nucleotide components. And there are nucleotides for DNA, and there are nucleotides for RNA, which are slightly different. However, both DNA and RNA nucleotides have the same basic components. The basic components you should know, and they are 5 carbon sugars, right there. In the case of DNA is deoxyribose. In the case of RNA is ribose. A phosphate group right here. A nitrogenous base right here. Okay, so notice that both group 2 and 3 are bound to the sugar. So the phosphate group is bound to the 5 carbon sugar. The nitrogenous base is bound to the 1 carbon sugar of the deoxyribose in the case of DNA, or ribose in the case of RNA. So make sure you know the components of nucleotides of DNA and differences between ribose and deoxyribose right there. And by the way, given a nucleotide, you should recognize that as a nucleotide. Okay. This cannot be an amino acid because it doesn't have the central carbon with the hydrogen and the nitrogenous group and the carboxyl group and then the R group. So that cannot be it. It has nitrogen, so that tells you it's not a carbohydrate, and lipids don't have hydrogens either, so it's not a lipid. Okay. Know that the nitrogenous bases come in two types, purines and pyrimidines. Purines are adenine and guanine. Pyrimidines are thymine, cytosine, and uracil. You should know those. These are the chemical structure of each. Purines are made of 2 rings. Pyrimidines are made of 1 ring. Let's see. Adenine and guanine are found in both DNA and RNA. Cytosine are found in DNA and RNA. Uracil is only in RNA. Thymine is only in DNA. And then go back to what you learned in Biology 1406 as far as the function and structure of both. DNA is a helix. It's where the genetic code is written. It's found in the nucleus of eukaryotes and the nucleoid of prokaryotes. The molecule is a the molecule has this backbone which holds the sugar and the phosphates, and then the base is to one side, which is the place from which the 2 helices in DNA come together. But all these will be part of another chapter, so don't worry too much about it. Okay. RNA is involved in protein synthesis. It's not a double helix, it's a single-stranded molecule. As far as antigenicity, it is large, it is huge. However, it's repetitive. All you have is a Lego tower with 4 pieces, 4 colors. That's not like a protein with a Lego tower with 20 different colors, or a complex carbohydrate, which could be repetitive, but then you add the varied colors of a protein or a lipid or something like that. This is pretty much very repetitive, so it's not antigenic. And then the other thing about nucleotides is that yes, they are made of they make up DNA and RNA, but I'm sorry. Okay, but okay. So let me go back. These are the building blocks of amino acids. I'm sorry. I'm a little bit distracted here. They're the building blocks of nucleic acids. However, individual nucleotides could also be made into high energy molecules. And the way that's done is, remember that the nucleotides we looked at only had 1 phosphate group. Notice that phosphate groups have negative charges on the oxygen. If another phosphate group is attached to that first phosphate group, the bond becomes unstable. Notice the squiggly line right there, meaning unstable, because now we have 2 negative charges close to each other, which are repelling each other, and it's not a stable compound. Now, if you then add even a 3rd phosphate group, now you have more negative charges all close to each other, wanting to leave very unstable bonds holding all this together. This is a high energy compound because it's unstable. This is what we call the readily available energy, the energy currency that cells use to proceed with chemical reactions. The most common nucleotide used for this purpose is ATP, which is adenosine triphosphate. You've probably heard of it. So you should know that adenosine triphosphate is a nucleotide. It didn't have to be ATP. It is, but it doesn't have to be. It could be GTP, CTP, TTP, UTP. Any of the other bases could be made into these high energy molecules, and the cell does that in occasions. Typically, though, it's ATP. All right, so you should know ATP, high energy molecule, is a variation of a nucleotide with two more phosphate groups attached. We talked about it not being antigenic. Okay, and lastly, lipids. And lipids are a little bit different in that there isn't a set building block. The only thing that, and this is a very diverse group, the only thing that binds all lipids together is that they are hydrophobic. That's the only thing, okay? However, all types of lipids essentially have the same components. They all have carbons, hydrogens, and oxygens. There is no nitrogen involved. We should know what lipids are made by: cell membranes, cell walls, energy storage molecules. There are two basic types of simple lipids, which are your carbon, hydrogen, and oxygen molecules, like triglycerides and sterols. And then we have compound lipids, which is a simple lipid with something else attached. And the one that we're going to look at are phospholipids. So simple lipids are made of a glycerol with 3 fatty acids attached to them. And the fatty acids can be, which are chains of carbons and hydrogens. They can be saturated or unsaturated. And this is an important concept. Yes, you could say this is important nutrition, complex unsaturated fatty acids are the ones that make oils. Saturated fatty acids makes things like solid fats like butter or lard. However, this is microbiology, okay? In microbiology, what do we need to know? Why do we care about saturated versus unsaturated fatty acids? The reason why we care is because a unsaturated fatty acid cannot become solid. It remains liquid because the kinks in the double bonds of the made by the double bonds in the carbons prevent these molecules from coming very close together. Therefore, this remains a liquidy towards the liquid type of consistency versus saturated fatty acids with the straight fatty acid chains will give you a solid consistency. So now you go back and say, okay, where do we find lipids in the cell? Okay, we find them in cell membranes. We find them in cell walls. Okay, what is the consistency of those cell membranes and cell walls? Is it liquid? Is it solid? And does it have unsaturated or saturated fatty acids? If it has unsaturated, then it's going to be liquid. If it has saturated, it's going to be solid. What does the cell need? And the answer is it needs both. But it does need for these structures to not be completely solid. They have to have a gel-like liquidy consistency to them. So back to this concept. But that is the reason why we care about saturated and unsaturated, is at the cellular level. How does that make a difference in the cell? Okay. So, so here we come to probably the most important lipid of them all, which is the phospholipid. It's a compound lipid. And the reason why this is so important is because we said lipids were hydrophobic, which is true, except that when you have a compound lipid, like phospholipid, now we have changed that a little bit, and we end up with a molecule that has a hydrophobic and a hydrophilic component. So now this is a very confused molecule. The molecule is made of the glycerol, the 2 fatty acids instead of 3, and 1 phosphate molecule. This is the difference right here, is the phosphate. Remember, phosphate always has the oxygens attached to it, and the oxygens are negatively charged. So here is the glycerol. Here are the 2 fatty acid chains, and then the 3rd carbon, instead of having a fatty acid chain, we have the phosphate group. The phosphate group has negative charges. So now we have a charged part of the molecule, and we have a hydrophobic uncharged part of the molecule. So this is the hydrophobic end, this is the hydrophilic end of the molecule. Notice they had the fatty acids, one is saturated, the other one is unsaturated, so that the consistency is going to be a fluid consistency, semi-solid fluid consistency. The importance of phospholipids is that they are the major component of cell membranes, both internal and external cell membranes. As a matter of fact, anywhere in this planet, in the living world in this planet, where there is a membrane made by a living organism, that membrane is made of phospholipids. So it's almost like the wheel. All wheels are round, all membranes are made of phospholipids. Okay? And the reason why is because of its hydrophobic and hydrophilic ends. If you throw phospholipids in a water environment, these molecules automatically, without the expenditure of energy or enzymes, will form themselves into a membrane shape, into a double layer membrane shape, so that all of the little heads, which are hydrophilic, will go towards the water, and the little tails, which are hydrophobic, will be in the middle of the molecule, creating this barrier, this hydrophobic barrier in the middle of the membrane, which can prevent things from entering the inside of the cell. However, the phospholipids must be fluid, so must have unsaturated fatty acids, so that the proteins, which are the channels and receptors embedded in this sea of phospholipids, can move about. So this cannot be a solid, hard shell. The cell would die. It has to be a fluid, imagine an oily shell, so that these proteins can swim about and change places and can actually go about their function. So you should know the components of phospholipids. You should know why they're so important. Being given a phospholipid molecule, you should know which is the hydrophobic and hydrophilic heads. And remember, oftentimes, we don't necessarily use these pictures in the PowerPoints. We may use the pictures in the worksheets. Okay. Other kinds of lipids are sterols, which these are carbon rings with a sterol OH group attached to them, a hydroxyl group attached to them. So there's the hydroxyl group. This is the steroid. The sterol is when the hydroxyl group's attached. These do not have fatty acids. These molecules have carbon rings. So they are going to be hydrophobic and they're going to be solid. So they have a solid consistency about them. What are they used for? In microbiology, in cells, in eukaryotic cells, sterols are positioned in the middle of the phospholipid membrane in order to give more consistency to this fluid, oily membrane. So it gives it a little bit more consistency. Oftentimes, there are cholesterol rafts that move proteins from different parts of the cell membrane. So they are extremely important in eukaryotic organisms. Prokaryotic organisms do not have sterols in their cell membranes, except for one genus of bacteria called Mycoplasma, which lacks a cell wall. And because it lacks a cell wall, its cell membrane does have cholesterol. And that would be important to know. Because this little genus lacks the cell walls, the cells don't have a definitive shape. So they have different shapes, rounds, rings, rods. There's not a definitive shape. Another group of lipids that's important in microbiology are called waxes. These are very water repellent, solid kinds of lipids that a specific genus of bacteria called Mycobacterium, not to be confused with Mycoplasma, keep those straight. Mycoplasma is the genus that has no cell wall. Mycobacterium is the genus that has a waxy cell wall. So the waxy cell wall protects the bacteria from anything entering the cell. And anything, it's imagine candle wax surrounding a bacteria, completely water repellent, solid, pliable if heated, but it prevents things from going in. Things like nutrients, which means these kinds of bacteria are going to grow painfully slow because nutrients will seep in very slowly. But at the same time, disinfectants, antibiotics will have a really hard time going through. So they will be very difficult to treat the infections with Mycobacterium. So it is important to know whether an infection is being caused by a Mycobacterium species. There are 2 species of Mycobacterium that are pathogenic, that are particularly bad, Mycobacterium tuberculosis and Mycobacterium leprosy. And we'll talk more about these cell walls in chapter 3. And the last type of lipid that we want to talk about that pertains to microbiology are a type of lipid called lipopolysaccharides. These are lipids with a polysaccharide attached to them. So this will be a complex saccharide, if you will. They're called, well, there's also unlipoproteins, okay, fine. But this is the one that we really want. LPS, lipopolysaccharide, is the component that makes the cell wall in a group of bacteria that we're going to call gram-negatives. And so we'll come back to these kinds of cell walls in chapter 3. So this is just as an introduction, an initial introduction to these terms and what are these compounds made of and where they're found. Lipids are not antigenic molecules unless they're attached to an antigenic molecule like a protein, in which case, yeah, they will become antigenic. All right, and that is it for chapter 2. Any questions? Hopefully I did address the question about antigenicity of these molecules, different macromolecules.